Good afternoon, everybody. Welcome you all to this Gyan Darshan session. Now, today I will focus on one of your courses, that is BBYCT 133, Plant Ecology and Taxonomy. In fact, I have covered some of the units of this course earlier also. Now, this is basically a course in botany, and it's a part of the BSc Botany CBCS and also the BSc Multidisciplinary program. So this is one of the courses in both of these programs. So there you have one unit, that is an introduction to the plant communities. Now, in one of my earlier Gyan Darshan session, I focused on another aspect of community, that is succession, which is basically a functional aspect of plant community. But today, we will be focusing mostly on the structural aspects of plant communities. I mean, what is meant by community, number one, and then if you have to study a community, if you have to study a plant community, then what are the different parameters based on which you can study the community? So first of all, let's start with what is meant by a community. Because we are dealing with the botany, so we will of course focus our attention here to the plant communities. What is meant by community? It is a group of several species living together with mutual adjustment and beneficial interactions in a natural area. So basically it's a collection of different species which is present in a particular area. And obviously the organisms which are present, it can be plant, it can be animal, they share the same habitat growing in a uniform environment. So basically it is an aggregation of living plants. And these plants, they have mutual relationships among themselves. Sometimes the relationships may be beneficial relationship. Sometimes it can even be detrimental relationships or antagonistic relationships. So you have the different types of interactions. When we are talking of a community, then we have the different types of interactions. Some of these interactions you might have already come across, like probably you have heard these terms like proto-cooperation, the amensalism, the commensalism, parasitism. So these are actually the different forms of interactions which are present in a community. So what is community? It's basically an assemblage of different species which is sharing the same natural habitat, and they are having some associations and they are having some interactions, whether beneficial interaction or some antagonistic interactions. Now, how this concept of community, how did it evolve, or what was the first instance from where this concept of community started? Initially, the term community was not there. Instead, there was a term called biosynosis. What is biosynosis? This is an association of organisms in an oyster bed community. So first, probably the concept of community actually came from this oyster bed community, and it was given a name called biosynosis. Later on, for the biological community, Carl Mobius in 1877, he actually extrapolated this concept to the different other plants or animals. And then these interacting organisms, they were described as a community. So the term biosynosis was then replaced by the term community. Now, whenever we are, when we are talking about community, you can well understand that we are actually dealing with so many plants and so many animals. So studying a community is not very easy task. Why? Because there are so many species. There are so many species of plants. There are so many species of animals. So how will you study? Now, for the sake of simplicity, we will describe, or for the sake of discussion, there has evolved different methods. Some of these methods, these were used in earlier times, and some of the methods, they are also used in the current research. Like if any ecologist basically wants to study the community in any forest or any other habitat, then what are the different ways, or what are the different index, or what are the different parameters by which the person, the particular ecologist can study? Now, initially, it was the floristic method. What is floristic method? It's a traditional method. Like you just go to a particular forest and then you study the species one by one. Try to know their genus, try to know the species, just as you do as a taxonomist. If you are a plant taxonomist, you go for an excursion to a forest or a hilly area, and then you try to study the different flora, okay? Then try to identify those flora based on the taxonomic characters, and then try to find out to which family do they belong, and then the genus and the species. But you can well understand that in a community, when you are doing this kind of following this method in a community, it is really cumbersome. Because in a community, you find so many, genus and so many species. And enlisting them one by one, by means of their morphological characters, floral characters, one by one, it's really tedious and it is a time-consuming process. So later on, for very large communities, this floristic method did not hold to be very much sound. So people needed some other kind of, methods or techniques. Then came number B, that is the physiognomic method. What is physiognomic method? Now this physiognomic method, this was actually given by one of a very well-known ecologist called Ronkear. And Ronkear actually what he did, he actually proposed the concept of life forms. I will come to this concept a little bit later. But just to introduce you right now, Ronkear actually said that all the plants, they can be actually categorized. Whatever plants you find on this earth, they can be grouped into 5 life forms. What are these 5 life forms? Phanerophytes, chemiphytes, hemicryptophytes, cryptophytes, and therophytes. So whatever plant you find, those plants can be categorized into any one of these 5 classes. Okay? We will discuss the characteristics of each of these life forms in short. So, like phanerophytes, cryptophytes, hemicryptophytes, whatever. So each of these life form, they actually coincide with a particular kind of climatic condition or a particular kind of habitat. So if you say that the percentage of phanerophyte is high in a particular habitat, you also get an idea about the nature of the habitat or the nature of the climatic condition of that particular habitat. And this is called as the biological spectrum. So biological spectrum represents the percentage distribution of species among the various life forms. Supposing you go to a particular area, you study the community. What you try to find out? You try to find out the percentage of each of these life forms. What is the percentage of phanerophyte? What is the percentage of cryptophyte? What is the percentage of hemicryptophyte? What is the percentage of therophytes? So you try to find out the percentage of each of these life forms. And based on that, you can also get an idea about the pattern of climate, about the pattern of soil, about the nature of the habitat, natural habitat. So through calculation of percentage value of each life form, the biological spectrum of the area is obtained. So I give you an example here. Let us suppose that on comparison of the biological spectrum of any area, with the normal biological spectrum of phanerogamic flora of the world, it is seen that the value of therophytes is higher in the spectrum of the area studied than the value of this life form in normal spectrum. For instance, I give you an example. Therophytes, the percentage of therophytes, they are very high in deserts. The percentage of phanerophytes, they are mostly higher in case of plain land. Like for example in the Indo-Gangetic plain, the percentage of phanerophytes is high. If you go to the desert area, there the phanerophytes are less, the therophytes are higher. As I told, that by looking at the percentage of each of these life forms, you will get an idea about the nature of the habitat. But if you find that in a particular area in the Indo-Gangetic plain, the percentage of therophyte is higher, it can happen, which is not the usual case. But sometimes you may find, supposing you go to an area in the Indo-Gangetic plain, you try to find out the percentages of life forms and you suddenly find that the percentage of therophyte is higher. Does that mean that it is a desert? No, it's a plain land. But then why the percentage of therophyte is higher? Because of some other disasters. It can be anthropogenic disturbances. You know that the different natural disasters that are being caused due to our reckless effects or our because of the intervention of the human kind or due to overgrazing. You know, the cattles, they feed on grasses, and because of this overgrazing effect, or maybe due to predator effect, or due to some infections, parasitic infections. So there, because of these reasons, or pest infestation, there you find that suddenly due to the other influences, biotic influences, the percentage of therophyte is higher. So this has been found to occur actually. And so we come across two terms, that is the normal biological spectrum and the what you actually see. That is the after calculation you may find that the normal biological spectrum you are not getting. There is a deviation. So this might happen. So what I mean to say that this concept of life form, this is very, very crucial. And what Roundkere gave this classification system of life form, that is very relevant because it has a lot of other significance as well. So from this if you have such a condition, then it indicates the deserty nature of the area, since abundance of therophytes is the characteristic of desert climate. And why this deserty nature? It can be due to different effects. As I said, it can be due to overgrazing, it can be due to infections, it can be due to the climate changes, okay, the global warming and the climate changes. So because of these kind of effects, you can get such percentage, abnormal kind of spectrum. So the biological spectrum does not take into account factors like biotic disturbances. Since the climate of Indo-Gangetic plain is suitable for forest development, ideally phanerophytes should have been higher. But in reality, it shows the dominance of therophytes. Why? As I already said, due to anthropogenic activities. Because of too much of human intervention, because of overpopulation, because of overutilization of resources, there is a depletion of resources. And that is why we are getting this kind of abnormal spectrum. So this might happen. Next we come to the quantitative characters of plant community. What is meant by quantitative characters? Quantitative, as you can well understand, that we are quantifying. We are trying to find out some numbers. We are trying to do some calculations. Obviously, they are some mathematical calculations. So what are those kind of parameters? Number one is frequency. What is frequency? Degree of dispersion in terms of percentage occurrence. So there can be some species which are widely spaced. There can be some species which are found in clumps or mats. Like you must have seen the bryophytes, the mosses. Okay, mosses, they mostly clump together. During the rainy season, you might have seen that the mosses on the brick walls and all, they clump together. They form mats. Whereas there are some other species again, which they can be, they can be widely spaced, and there are some species which are again, they form kind of clumps. Okay, so that is actually meant by the frequency. Distribution patterns indicate their reproductive capacity and their adaptability to environment. So again, Roundkere recognized 5 frequency classes based on low frequency and high frequency. Low frequency 1 to 20, 21 to 40, 41 to 60, 61 to 80, 81 to 100. Accordingly, he has given 5 categories, that is A, B, C, D and E, based on the percentage of frequency. Next comes density. What is density? Whenever we talk about density, that means per unit area. We are calculating the number of individuals of a particular species per unit area. So density gives an idea regarding the numerical strength of a species in a community as well as the degree of competition. In some cases, as grasses and vegetatively propagated plant, the term individual creates difficulty. So if you think of grass, right, if somebody says, how many grasses are there? It's very difficult to say. Okay, if somebody says how many mango trees are there, you can calculate it very easily. But if somebody says how many grasses are there in this particular habitat, it's very difficult to calculate. So what happens? How will you calculate in this case? Supposing a grass species, you know about the Gramineae or the Poaceae family, which has mostly the grass. It's called the grass family. So supposing you want to study the grass species in a community, it is not very easy to calculate how many such species are there. So in that case, what you have to do? Each aerial tiller or shoot arising out from the soil, that is regarded as one individual. Okay, so that is how you calculate the density. Relative density, the proportion of density of a species to that of stand as a whole, that is relative density. Next, what is abundance? Abundance means the total number of individuals of the species in all the sampling units divided by the total number of sampling units in which species occurred. What are sampling units? Sampling units means when you go to a forest, it may not be possible for you to study the community in each and every corner of the forest. So in that case, what you have to do is you have to make certain sampling units. Like you choose a particular small area, and that area is used as a sampling unit to get an overview or to get a holistic picture of the community of the forest. And these kind of sampling units, in ecological terms, they are mostly of 2 kinds, which are known as the quadrate and transect, which today I am not discussing. So quadrate basically is a rectangular plot of land. I mean, you can select a sampling unit in the form of small rectangles or in the form of small squares. I think it is better to say that square sample, and then find out what are the communities within that sampling unit. Once you find it out, you extrapolate it. Okay, like we study a small sample, and then like we study a small sampling, make a small sampling unit, and we study and then try to make a prediction about what would be the overall, picture of the overall forest. So that is actually meant by the sampling unit. Okay. So again, you have the abundance classes, that is rare, occasional, frequent, abundant, very abundant. So you take per square meter quadrate, that is the sampling unit, and you find out how many individuals are there. A particular species, how many individuals of a particular species are there. So whether you call that species as a rare species or whether you call it as a very abundant species. If it is 100 plus, then it's very abundant species within a particular of course, sampling unit, that is quadrat, per square meter of a quadrat. Next we come to cover and basal area. What is cover? It refers to the area of ground occupied by the aerial or above ground parts. What are aerial parts? Leaves, stems, flowers, these are all above ground parts. That is not the roots, leaving the roots, the other parts which are above the ground. So how much area is covered by these plant parts? That you have to calculate. So obviously you have to take a view from the top. And from the top you take a view and see that what is the area covered by the aerial or the above ground parts. So that is called as the cover. What is basal area? Basal area is just the reverse. That is the ground actually covered by the crowns or by the stems penetrating the soil. So it is more close to the ground. It is not the aerial part. It is more, basal area actually refers to those which are more grazing the ground. In forest, the basal area is the cross-section area of a tree measured at 4.5 feet above the ground. In case of grasslands, basal area refers to the coverage of ground 1 inch above the ground surface by stems and leaves. Mind that it is not the underground parts. It is basically the leaves and stems. But leaves and stems which are not very emergent. Leaves or stems that are almost grazing the ground surface. Like for example, radish. Here the leaves you will find that they are these kind of leaves. They more or less graze along the surface. So this is one of the examples. Next, basal area per tree is equal to the total basal area divided by the number of trees. The area of coverage is used to express dominance. Higher the coverage area, greater is the dominance. So cover is actually an index to measure the dominance, because if a plant is dominant, if a species is dominant, then the cover will also be large. It will cover a large area. So high cover means the plant species is more dominant. Now another very important parameter we call association index and index of similarity. What is this concept? Supposing you are studying 100 quadrats. I am just giving you an example. Supposing you are studying 100 quadrats. I think by this time the concept of quadrat is clear to you, that it's basically a sampling unit. So out of 100 quadrats, you find one species A in 90 quadrats. Another species and species A in association with another species B is found in 40 quadrats. So basically you are studying the species A. You find species A in 90 sampling units. And species A together with species B is found in 40 quadrats. So if somebody asks you what is the association index of A, then you have to put this formula. Total number of quadrats in which species A occurred in association with B divided by the total number of quadrats containing A alone. Obviously it will be 40 divided by 90. So it comes around 0.44. So in this way you can calculate the association. So association index will also give you an idea regarding whether the species is prominent or not. Okay? Because not only you are studying in one sampling unit, not only you are studying one particular species, okay, in the quadrats, but how much that species is associated with another species. So that will also give you an idea regarding how common or how commonly occurring is that particular species. So in that way, this is a very important parameter in a community study. Now, suppose in one group of existing species, the number of plant species is 30, and in another group, the number is 20. In the first and second group, 15 species are common. Then what is the index of similarity? You have to use this formula. Twice the number of common species divided by total number of species in both the associations into 100. Basically, it's a percentage. That is 2 into 15 divided by 30 plus 20. So, multiply it by 100. So, it gives you 60. So, basically, its index of similarity is basically a kind of percentage study. Next comes importance value. The overall ecological importance of a species in relation to the community structure can be obtained by adding the values of relative density, relative dominance, and relative frequency. So, density, dominance, frequency, all these terms have already been introduced to you now. And now, if you add these values, then what you get is the importance value. Okay. So, importance value is usually you assign each of these parameters 100 points. So, normally your index value or this importance value, that is importance value index, it comes in 300. Okay. So, 100 plus 100 plus 100. So, that is total 300. This is known as the importance value index. Now, IVI gives complete picture of sociological character of a species in the community, but does not give the dimension of relative density, relative dominance, and relative frequency. So, IVI is clear. IVI means that is the summation. And how do you represent this IVI? You represent it in the form of a phytograph, which is called as the Lutz phytograph. How does it look like? It looks like this. That is, a circle is made and it is divided into 4 equal parts by 2 radial lines lying at right angles to each other. Divide the 3 radii from center to circumference into 100 equal parts and the 4th radius into 300 parts. So, one radius 300 parts, the other 3 100 parts. Mark the first 3 radii with RD, RDM and RF. That means relative density, relative dominance and relative frequency. 4th will represent IVI, that is importance value index. So, this is how you actually see this photo or the diagram. So, you can get an idea about how a phytograph actually is represented. Okay. Next we come to, so these are the quantitative characters that I discussed. Now we come to the qualitative characters. Qualitative characters, we will start with the physiognomy, phenology and these type of characters. What is physiognomy? Physiognomy means the shape, stature and the growth form of the dominant species. Phenology, that is the scientific study of seasonal changes. You all know that the season, it has an effect on the appearance and disappearance of plants. You see certain plants which are predominant during the spring season. You see certain flora which are predominant during the rainy season. So, the seasonal effect, that is termed as the phenology. Different periods of seed germination, vegetative growth, flowering, fruiting, leaf fall, seed and fruit dispersal. So, this is dependent on the seasons. And these events are shown by the phenograms, and the study of date and time of these events, that is different physiology like flowering, dispersal, leaf fall, fruiting. If you study these kind of physiological processes based on season, you call it phenology. So, obviously, in a community study also, phenology is a very important component. Third point, that is stratification and aspection. This is also very interesting parameter. Because when you go to a forest, like huge forest, you must have seen that there are huge trees. There are some understories. Okay? That is the plants which do not grow to much height. They prefer to live in shade. Ferns, for example. They live in the shade of the very large trees. Then there are the different kinds of mosses, the algae, the bryophytes. They prefer the shady places. Okay, so there are different height plants. Some are understory plants. Some form a canopy. Some are medium. Some plants are moderately tall. So you have some kind of layered structure. That is a stratification. Strata means layers. Stratification means you have a layered pattern of floral distribution. So the way in which plants of different species are arranged in different vertical layers to make full utilization of the available physical and physiological requirements. Now this is also very interesting, that why the plants, they choose to be of different height. I mean, we look at plants in a way that they are just randomly distributed. But is it really random? No. The plants are equally intelligent. They can equally assess that how they can utilize the resources to the maximum. If all of the plants, they would have grown to the same height, would they survive? The answer is no, because then there would be a tremendous competition between them for the sunlight, for the minerals, for the nutrients. So if you have to make a community, then there should be some kind of sharing of resources. And the plants equally judge that if we are to make a community, then we have to be helpful to each other rather than trying to be enemy or detrimental to each other. Then only we can form an association. Then only we can form a community. If we interact in a beneficial way, if we help each other. Because if they do not coexist, they cannot form a community. They will be individual. And that is probably one of the reasons that the plants have chosen to remain in layers, so that there is no acute competition. Some which are shade-loving plants, they will not compete for sunlight. Some which are very tall trees, they will use the solar energy. Maximum utilization of solar energy. Okay? So, if one periodically observes the flora for the whole year, changes in the appearance of vegetation, it is apparent with the change in season. So, seasonal changes. Vegetation change due to seasonal change. This is known as aspection. And stratification, I already told you that the different community, different individual species of a community, they are not present to the same height. They are present in different layers or strata. So that is called as stratification. So number of layers is distinguished. L1, ground stratum, mosses, lichens, thallophytes, algae, fungi. L2, the ground flora. L3, shrubby layer. L4, canopy layer of trees. So you can see that they are present in layers. This is called stratification. Next is sociability. Sociability, what does it mean? It denotes the proximity of plants to one another. How close the plants are. We say that ants are social animals. Why? Because they live in a colony. They live close to each other. So the same concept, sociability. Abundance, I have discussed that plants are not found uniformly distributed in an area. They are found in smaller patches or groups, differing in number at each place. Mostly I gave you examples like the mosses. the hepatics, like Riccia, they form, they form a rosette. Or mosses, I gave you idea. Then lichens, these are present in patches, they are present in groups. So abundance divided in 5 arbitrary groups: very rare, rare, common, frequent, and very much frequent. Next is interspecific associations. How is this association possible? It is possible if species can live in similar environment, species have similar geographical distribution, species belong to different life forms, plants of one species are related to plants of other species. So we call them as interspecific association. Now we come to in detail, that is life forms. Roundkear, I already introduced you that Roundkear employed 3 guiding rules in selection of life forms. I mean, what was the basis of forming this life form? The character must be structural and essential and must represent morphological adaptation. Character must be sufficiently clear that one can see in nature. All the life forms must constitute a homologous system and must enable a comparative statistical treatment of flora of different communities. So these were the 3 main points based on which Roundkear characterized the 5 life forms. And these are named as, I have already told, phanerophytes, chemiphytes, hemicryptophytes, cryptophytes, and therophytes. Now we come to the definitions of each of these. What are phanerophytes? The perinating organs, which include buds, they are present on erect shoots present above the ground. So buds are present much above the ground. These buds are naked and exposed to external environmental condition. So phanerophytes include trees, shrubs, and climbers present in the tropical climate. Then comes chemiphytes. Chemiphytes, the perinating organs are present on shoots found close to or just above the ground. So here the perinating organs are not that high. They are present just above the ground. That is the difference. The buds receive protection from external environmental condition by fallen leaves and snow cover. But in phanerophytes, buds are not protected because they are much above the ground. But here the buds are protected. Example, creeping woody plants and herbs found in cool dry climate, Arctic and Alpine region. So chemiphytes are mostly found in Arctic and Alpine regions. Phanerophytes in the tropical regions, mostly. Hemicryptophytes, perinating organs are present near the ground and receive protection from fallen leaves and soil cover. What is the difference between chemiphyte and hemicryptophyte? In chemiphyte, the buds are present just above the ground. But here, the buds are present near the ground. It is more close to the ground. Biennial and perennial herbs growing in rosette. These are found in cold, temperate zones. Cryptophytes, the 4th life form. The perinating organs or buds are buried in the ground. What is the difference? They are below the ground surface. That is, they are present under the soil cover. They are covered by the soil. And why they are covered by the soil? Because they are being protected from freezing or drying. Like mostly in the arid regions. The tuberous or bulbous herbs mostly. Finally, the therophytes. These are annual plants and complete their life cycle in summer or favorable season. So they complete their life cycle within a very short time when the rainfall is there or when abundant rain is there. Seeds, which are the perinating organs, remain dormant during unfavorable season. They do not germinate. Only with the availability of the rainwater, they germinate and they complete their life cycle within that short span. Mostly the desert plant, the grasslands. This kind of community they are formed by the therophytes. So these are the main 5 life forms. So you can see that based on these life forms, you get an idea about the zone. You get an idea also about the climate of a particular region. Now, what is the normal biological spectrum? That is, they have shown phanerophytes 46%, chemiphytes 9%, hemicryptophytes 26%, cryptophytes 6%, and therophyte 13%. Phanerophytes, tropical rainforest, it can be as high as 74%. Therophytes above 40%, desert climate. Chemiphytes above 50%, extremely cold climate. High percentage of hemicryptophyte in temperate forest, extensive grassland. So you see, one is normal biological spectrum and another, as I already mentioned at the beginning, that what is the biological spectrum that you are getting. If you go to a desert, you will not get the normal biological spectrum because obviously the therophytes will be higher. If you go to an extremely cold climatic region, you will not get the normal biological spectrum. You will get the chemiphytes to be very high. Okay, so one is just a normal or an average which more or less you can find on this earth, and another is based on the region, these percentages will fluctuate, from which you can get an idea about the nature. But sometimes it can be an anomaly, as I already told. In Indo-Gangetic plain, the phanerophytes should have been higher, but somehow you are getting therophytes to be higher. Because why? Because Indo-Gangetic plain is a very densely populated region. And therefore, due to the human intervention or anthropogenic activities, our reckless effects or our reckless activities, you are not getting the phanerogams to be higher or phanerophytes to be higher. Rather, you are getting the therophytes to be higher. So these are some anomalies. Otherwise, it follows a particular pattern. Next is vitality. That is the capacity of normal growth and reproduction, important for survival of species. In plants, stem height, root length, leaf area, leaf number, and weight of flowers, fruit, seeds, they determine the vitality. And again, you have the vitality groups. Mishra and Puri in 1954, they have actually given vitality groups. That is V1, V2, V3, V4, and V5. So 5 groups. V1, seedlings are highly vulnerable. V2, seedlings grow but unable to reproduce. V3, reproduce through vegetative propagation. V4, reproduce sexually but uncommon. And V5, reproduce sexually and grow regularly. So these are the 5 vitality groups. Now we come to synthetic characters. One is fidelity. That is degree with which a species is restricted in distribution to one kind of community, also known as indicators. So one particular species, there are many species which are restricted to one community only. They are not very much, dispersive. So that is called fidelity. Presence, extent of occurrence of individuals of a particular species in a community. That is how uniformly a species occurs in a number of stands of the same type of community. Again, rare, 1 to 20%, seldom present, 21 to 40%, often present, 41 to 60%, mostly present, 60 to 80%, and constantly present, 81 to 100%. What is constance? This is the degree of presence. So this presence and constance, these two terms are related terms. The degree of presence in a unit area. So if you measure the presence in a unit area, it becomes constance. So here you are studying a sampling unit, that is unit area, and not the whole stand. Then we call it a constance. So it improves the method of presence study. Again, constance 1, 2, 3, 4, 5. You have these 5 groups. So 1 to 20%, 21 to 40%, 41 to 60, 61 to 80, and 81 to 100. Again, they have given the 5 groups. Now we come to a very important concept of community, that is ecotone and age effect. What is meant by ecotone? Now, you go to a forest or any kind of community, and you try to find out two communities. Supposing you want to study two communities in a forest. Is it really possible for you to demarcate between two communities in a forest? Answer is no. Where does one community ends and another community begins? I am talking of plant community. Where does one community end and another community begins? It is very difficult. It is very difficult to mark a demarcation. It is very difficult to mark a transitional zone also. But ideally, there is a transition zone. So, in reality, there is never a distinct boundary, or you cannot imagine a sharp point or a distinct line that demarcates one community from the other. That is, here is the end of one community, here is the beginning of another community. No, that is difficult. Rather, two or more different communities, they meet together. They integrate or they integrate together in a transition or a marginal zone. That transitional zone is called ecotone. So you cannot make out which is the end of one community and which is the particular point that is marking the beginning of another community. Rather, there is a transitional zone where one community ends and another community begins. And that intervening zone is called ecotone. And surprisingly, it is found that in this ecotone or in this transitional boundary, maximum number of species, they actually flourish. Or maximum number of species, they represent the intermixing of species of both the communities, and that is why they are very rich in species. So ecotone has a very, kind of ecological significance. The transition zone, like for example, transition zone between grassland and forest. The intermediate zone between any two major land or aquatic communities. Ecotone has sufficiently greater number of species and density of most of the species is higher than that in neighboring or bordering community. And we call it principle of ages or age effect. What is age effect? Age effect is that ecotone region has the greater number of species. Because probably, why? Because probably it is enjoying the ideal conditions of both the bordering communities. Because that is the transition zone. So this zone is really very significant. A common example of age effect can be seen in those species of owl that live in or near ecotones between forest and grasslands. They depend on forest trees for nesting and do their hunting in grassland, where they depend on field rodents for food. The age effect is chiefly due to wider range of suitable environmental conditions. Ecotone is colonized by species that are commonly found in the communities on both sides, as well as by some versatile species of plants and animals. So you can actually give many more examples on ecotone. So these are just few examples. Now, extent of ecotone is variable. If somebody asks you whether the ecotone is narrow or whether the ecotone is broad, whether it is wide or whether it is narrow, It can be both actually. So when you are talking about ecotone between a pond and an adjacent upland or heavily grazed and ungrazed areas, the ecotone is narrow. But if you talk of evergreen forest and deciduous forest, the ecotone is wide. Therefore, some ecologists have introduced the continuum concept, which means that there are no distinct communities with well-defined boundaries. But there is a gradual change in space and time along some sort of gradient, moisture, temperature, soil, and such gradient is very, it's very common in plant. That is patchy resources. Even in the same tract of land, you may find some areas where the resource is abundant and some areas where the resource is scanty. So we call it a patchy resource. That is, the nutrients are present in patches. They are not evenly distributed. And the plants actually face such situation. Of course, the plants make different modifications by which they try to reach more towards the high resource areas where they can actually do more profuse rooting and they can grow abundantly. So this is known as the boundary, or this is known as the gradient effect. So that is very common, and that has to be taken into consideration when you are studying a community. Now another very important concept, that is dominance index. Dominance index is given by this formula, that is summation of Ni by N. Ni, importance value of species in terms of number of individuals or biomass, and capital N, total of corresponding importance values of all the component species. I think importance value, you have got the idea, I have already discussed. And similarity index S is equal to 2C by A plus B, where S is the similarity index. A and B are the number of species on two different communities. C is the number of species common to both the communities. And dissimilarity index, that is D equal to 1 minus S. So based on this, a very important formula has been given, and that is called species diversity, which is given by the Simpson's diversity index, which is given by this formula, that is D is equal to 1 minus summation of pi square. Now I give one example to explain this formula. Let's say you have a community where you have 5 species, A, B, C, D, E, and you calculate the number of species, each of these species. Now I ask you, what is the Simpson index value for this community, based on which you can calculate the species diversity. Straight away you put this formula. You count take the summation of the individual species, which comes to be 202. Then you calculate PA, okay. So this formula, once again you look at this formula, that is P squared. So basically we are calculating we are calculating the P squared. But how will you get this P value? Individual number divided by total, 104 divided by 202. Then for P, small p, 19 divided by 202. So you get this values, and then you put it square, whole square. 1 minus individual whole square. Okay. So when you subtract it, that is 1 minus 0.4, you get 0.6. So this is how you actually calculate the Simpson's diversity index, which gives an estimate of the species diversity. Now towards the end, I raise another problem, like if somebody asks you a question like, do communities, they have a tightly prescribed organization, or it is just a random assemblage? Clement said that it is very kind of tightly regulated phenomenon. That is, communities are stable functional units. If you remember, Clements also gave us the monoclimax theory. So communities are stable functional units with a fixed composition. Gleason said that no, communities are unstable and variable. They are more kind of random assemblage. So there is this dispute, and in order to solve this dispute, one experiment was conducted which is called kiddie pool experiment. It's a very simple experiment. Jenkins and Bikema, they conducted an experiment to see whether artificial ponds would develop predictable assemblage or random assemblage. What did they do? They set up 12 identical ponds filled with sterile water. Came back in a year to study the composition of resulting communities. What was the result? The ponds had very different composition of species. Accidents of dispersal and different dispersal capabilities affected which species ended up in each pond. So it was not at all an ordered kind of thing. It was very erratic. 12 different ponds are having 12 different kind of assemblage of species. Pattern is totally different. If it was a uniform phenomena, then all the 12 ponds would have shown the same kind of species assemblage. But in reality, it was not that. So Gleason's view was supported. That is, composition of community is dictated largely by chance and history. It is a random phenomenon. So that is the significance of this kiddie pool experiment. I think the last part we have already discussed. Even then, I just repeat that vertical stratification in a forest community. Like in a subterranean zone, forest floor, ground vegetation, extending a meter or so, understory tree and shrub layer, tree layer or top story. So there can be different kinds of, different kinds of combinations or different kinds of organization. In a grassland community, there are 3 strata: herbaceous layer, ground or mulch layer, and the root layer. So when we are talking of horizontal stratification, there are 3 different types of distribution. Random, each organism appears to be placed without any regard to where another organism occurs. No negative or positive interaction. Clumped or contiguous, that is, several individuals are clustered together in various spots. Ants into colonies, we talked of moss, the hepatics, the liverworts, and regular or uniform, that is produced due to negative effect of one organism on another. Each organism is spaced at certain intervals from others in the same species. That is, it's a very uniform kind of pattern. So today what we have done is basically we have focused on one of your units in plant ecology and taxonomy. Just to summarize that there is a topic or there is a unit called Introduction to Plant Communities, and we have tried to look at the different characters. We have tried to define the community, and then we have tried to look at the different characters of the plant community, right? First of all, the different methods of study of community, and then we tried to find out the different characters, right? How will you study a community? Is there any ordered kind of, characters based on which you can study the different species in a community? Okay, and the ecotone effect, that is, when you are studying 2 adjacent communities, then where should you focus upon? Which is the zone of ecological significance. And Round-Kier's life form, this is also one of the very important concepts. The definitions of each of the life forms and their characteristics. So go through the unit. If you find out any difficulty or you need any clarifications, you can write to us or feel free to ask questions. Okay, so please go through the unit, and we will come back again in some other session with a new topic on the same course. Thank you all, and a very, very happy learning.